All right, your big exam on molecular genetics is right around the corner, and we are going to make sure you are ready. So over the next few minutes, we're going to hit all the absolute must-know concepts from the DNA blueprint all the way to the ethics of gene editing, so you can walk into that test and just crush it.
Okay, so here's our game plan. We'll start with the master blueprint, DNA, and then see how it actually builds proteins. And we're going to spend a little extra time on how genes get controlled. Seriously, this is a huge topic for your exam before we jump into mutations, modern biotech, and the major ethical questions that come with all of it. Let's dive in.
All right, to really get a handle on genetics, you got to start at the beginning with the master blueprint itself. This is the fundamental instruction manual for every single living thing on the planet. Which leads to this massive question, right? How does life actually store its instruction manual? I mean, for decades, this was one of the biggest puzzles in all of biology. And the answer, well, it changed everything.
And here it is, the answer. Deoxyribonucleic acid, or as we all know it, DNA. Now, we all know the iconic double helix shape, but the real magic? It's in the sequence of those four chemical bases, adenine, thymine, cytosine, and guanine. That sequence, that ATCG pattern, is the code that writes the entire story of life.
Okay, listen up, because this is a 100% guaranteed exam topic. You absolutely have to know these three key differences. So DNA, the master blueprint, has deoxyribose sugar, it's that classic double helix, and it uses thymine. Its trusty messenger, RNA, uses ribose sugar instead, it's just a single strand, and it swaps out thymine for a base called uracil. Seriously, burn this slide into your brain, it's pure gold for your test. And check this out. This shows how DNA makes a perfect copy of itself. The process is called semi-conservative, which, is just a fancy way of saying that when DNA unwinds and copies itself, each new molecule is made of one of the original old strands and one brand new strand. It's an elegant system that ensures the code gets passed on accurately. Okay, so we've got the blueprint. Now what? How does the cell actually read that code to build all the proteins that do all the work? Well, that brings us to what's called the central dogma. This whole flow of information is known as the central dogma of biology. And honestly, this analogy just makes it super easy to remember. It's a simple two-step process. First, you copy the recipe out of the main cookbook, and then you take that copy into the kitchen to actually make the cake. And for your exam, you absolutely have to know the what and the where for both of these steps. So transcription is step one, copying the DNA into a messenger RNA molecule. That happens safely inside the nucleus. Then translation is step two. That mRNA message travels out into the cytoplasm, finds a ribosome, and it's read to build a protein. Simple as that. All right, let's stop for a second because this next section, this is a massive concept for your exam. I'm talking about gene regulation. I mean, think about it. Every single cell in your body has the exact same set of DNA instructions. So how does a brain cell know to be a brain cell and not a liver cell? How does it decide which instructions to actually use? And it really all comes down to gene regulation. The best way to think about it, it's like a bunch of light switches. Your skin cells and your muscle cells have the exact same giant cookbook of DNA, but they read different recipes by flipping different genetic switches on or off. That selective control, that is everything.
And here's what that switch actually looks like on a molecular level. You have these special proteins called transcription factors that bind to the DNA. Some of them are activators. They're like the on button, helping to start transcription. And then you've got repressors that basically block the machinery, turning the gene off. This pretty simple on-off system is what allows for all the incredible complexity of life.
Okay, so far we've been talking like the code is perfect. But what happens when there are typos? Well, that's where mutations come in. Okay, first things first. It is absolutely crucial to get this. A mutation is just a change. That's it, a change in the DNA sequence. I know we tend to think of mutations as being, bad, and yeah, some are, but a lot of them do absolutely nothing. And some can even be beneficial. They're the raw material for evolution. So this table right here breaks down the three main flavors of point mutations, which is when just a single DNA letter gets swapped out. A silent mutation? No effect at all. A missense mutation changes one amino acid, which could be a big deal or not. But a nonsense mutation? That creates a premature stop signal, which usually has really serious consequences for the protein.
But as bad as swapping one letter can be, deleting or inserting one, that's often way, way worse. We call this a frame shift mutation,
Because if you mess up the reading frame, every single three-letter word that comes after the mistake is completely scrambled. It turns the entire rest of the genetic message into total gibberish.
So we've seen how the code can change all by itself, but now, let's talk about how we've learned to get in there and mess with the code ourselves. Welcome to the world of biotechnology. So one of the most powerful tools in all of biology, hands down, is PCR, the polymerase chain reaction. Honestly, the easiest way to think about it is as a molecular Xerox machine. It lets scientists take one tiny microscopic piece of DNA and make billions of copies of it in just a couple of hours.
This diagram gives you the 30,000-foot view. The whole process is really just a cycle of heating and cooling that forces the DNA to copy itself over and over and over again, leading to this incredible exponential growth. So let's break down exactly what's happening.
And here they are, the 3 steps you absolutely have to know for the test. First, you denature. You crank up the heat to about 95 degrees Celsius, and that separates the 2 DNA strands. Then, annealing. You cool it way down so these little guide sequences, called primers, can attach to the DNA you want to copy. And finally, extension. You warm it up a bit, and this amazing heat-proof enzyme called Taq polymerase starts building the new DNA strands. You just repeat that cycle 30 or 40 times, and boom, you've got billions of copies. You know, this incredible power to read and copy and even rewrite the very code of life, it leads us to our last and maybe our most important topic, the ethics of genetics. With this kind of power comes a whole new world of really profound questions. You know, the Human Genome Project was just monumental, an incredible achievement. But it also kind of opened a Pandora's box of really complex issues. People often call these ELSI. That's for ethical, legal, and social issues. We are now forced to deal with questions our ancestors couldn't have even imagined. And look, these are not some far-off abstract ideas anymore. I mean, really think about it. Who should get to see your genetic information? Your insurance company? Your boss? How do we stop people from being discriminated against based on their genes? Where do we draw the line on editing the human genome in a way that could be passed down for generations? This isn't science fiction. This is the reality we're dealing with right now.
And that really brings us to the biggest question of all. We've developed the ability to fundamentally rewrite the code of life. But just because we can, does that mean we should? The challenge for your generation is going to be finding the wisdom to use this incredible power responsibly. It's definitely something to think about long after you've aced this exam. Now go get 'em. Good luck.